G'day, my name's Ben Newsom from Physics Education, and I am so excited to show a real space science project that you can get your students involved in October 2022. This is called the ExoLab, and it involves schools right around the globe with devices just like this, growing legumes, growing red clover, and seeing if they can get it to nodulate with a bacteria called rhizobia. Why? Well, that same device is on the International Space Station. In fact, it's just come down back from the ISS at the end of September 2021. And we are looking at the nodules in these ground trials and comparing them to, well, the nodules that were in microgravity for nearly a month on the ISS. The reason that we're looking at this is that we are at some point going to go to space to live, to grow things, to hopefully survive without needing resupply missions all the time. This particular device is one of the many experiments that's involved with working out how do we grow plants in space? How do we get plants to grow on the regolith of Mars or on the moon or in microgravity on a space station so that astronauts can actually have nutritious food? It's really powerful. In fact, I want to go a little bit closer to show you what this device kind of looks like. You can see the legumes that are still growing after a month in there, actually. And in this case, you'll see there's some lights and stuff in the little display. This is an Internet of Things device. It connects live to schools all around the globe, and you can compare the luminosity, the temperature, the carbon dioxide, and the humidity, not just what's happening inside here, but on the ISS as well. There's a tiny little camera in here that takes little shots and actually records the growth over that month, and you actually have exactly the same feed coming from the International Space Station as well. It is such an amazing opportunity for students to get involved in. And what we're going to do is going to pass you over to Laurie Waters, who can describe why on earth is the ExoLab project so important and really how can you get involved? And I must say the easy way out of that is get in touch with us and we can get you all the information that you can know about for October 2022's launch up to the ISS for ExoLab 10.
So our goal with ExoLab 9 is to achieve root nodulation during the stress of space flight. Tony did a great job designing our mission patch this year, and so he can definitely get you those files as you need to include that in presentations and such. Here's our leadership team, who I think you've met, all of us. I think you might have gotten a chance to meet Michael Wilkinson at some point during one of them. And if you haven't, then he will be on some of our future calls and our Legumes in Space live events that we do. He is my co-host for those. And our co-investigators and our curriculum development team, these are teachers around the world. They teach both in formal classroom environments as well as in informal settings, a really great mix. And our most recent addition to this team was Lisa Turney. She was selected as our ExoLab 8 Astromoji mission specialist, and her Bitmoji is the one that was selected to go with the ExoLab 8 mission to the International Space Station. And so in the video that you see of the astronaut powering down and removing the Cube Lab from the locker, her Bitmoji is on there and giving a little shout out to her school in Linwood, Kansas. So we have flown 8 missions with our flight provider is Space Tango. And so we've had a mix of different plant experiments is primarily what we've flown. There has been a bacteria experiment or a combination of plant with bacteria, microgreens, as well as the model organism Arabidopsis. So if you dig into plant research for space, you will often come across results for and papers written about Arabidopsis. It has pretty much been the model organism, but NASA and other researchers are branching out and really exploring other plants to grow. Currently, there's the chili peppers challenge is going on, and one of our friends over at Kennedy, Jacob Torres, got a lot of citizen scientists involved in that. So they're growing chili peppers in the advanced plant habitat. They grow microgreens as well, and that's something that we did in a prior ExoLab mission. And we did a big challenge at the end of last school year, of leguminate challenge, which plant should fly. And so students across the country at the beginning of that pandemic during fourth quarter of the US school year, we put this challenge out to teachers and students saying, which one should it be? And ultimately we decided on red clover to be the flight specimen for 8, and we decided to continue on with that plant for ExoLab 9. It is a replication mission of sorts for ExoLab 9. We flew 9 seeds during that mission, and we had some engineering challenge as well as some science challenges. It was a demonstration project on both fronts. And so we said, you know what, we want to do this again and see if what results we get. We haven't completed all the post-flight microscopy and science investigation for 8, so that is still underway. We're really focused on cultivating this next generation of scientists, engineers, and leaders through these STEM experiences and emphasizing curiosity. So we want really kids to get engaged through experiential learning and to ask more questions. We can give them this experiment and say, here are some of the variables that we are testing, but what other questions do you have? And we want them to develop those scientific inquiry and critical thinking skills. So we do this through project-based learning. And so it's mostly been K-12 for our focus and learning about astrobotany and the scientific inquiry process. We do have some college students who have expressed interest, and so we are looking at expanding into undergrad. I'm a graduate student myself, and so I have used a lot of this in my research and my projects for graduate school. So scientific inquiry, conducting those ground trials, growing plants in their classrooms, or this past year in their kitchens or dining rooms or bedrooms or wherever they could, gathering recyclable materials, as well as using our student kits. And they grow plants at home, and then they're able to compare the data of what they see in maybe their classroom ExoLab, as well as their student kit, and what they've seen on the ISS. As I mentioned, our science mission goal is to achieve root nodulation by the symbiotic rhizobia bacteria on red clover plants while under the stress of spaceflight. And that's never been done before. We're the first ones to try this. So we are going up on SpaceX 23, which is a cargo Dragon mission, on August 29th, and returning on SpaceX 23, cargo Dragon, on September 29th. You'll remember maybe from ExoLab 8, we went up on Northrop Grumman flight, the SS Katherine Johnson, and actually today I saw on Twitter that that Cygnus has been released from the station. But unlike the cargo Dragon, it does not return to Earth. Well, it does return to Earth, it just doesn't make it all the way to the ground.
In a fiery ball.
It goes in a fiery ball. So right now, the Dragon is our way up and down. So if we ever go up on a different type of flight, at least for right now, we have to wait for a Dragon, either with the astronauts or for cargo to come home. Hopefully that will change as other companies are approved to reach the station. I think Boeing Starliner is still out there trying to get their approvals. So, but we're going up on SpaceX again. So the lessons, particularly for ExoLab 9, are going to emphasize the scientific inquiry process. I don't know how your teachers may do it in Australia, but from a US perspective, it's very common with just about every grade level at the beginning of the year to review and add on, go a little bit more in depth with scientific inquiry A lot of students and schools participate in school science fairs or student science projects. And so they use this throughout the year with conducting investigations and experiments. So teachers often lead off with it in some way. So it'll be a good fit for really going in depth through each part of the process. Even the youngest ones in elementary school, teaching them how to ask a scientific question. And then even with our upper school, high school students, we find that we still must review with them every year what is an independent and a dependent variable, because somehow they forget. That summer slump time not only affects their math abilities, but we have to review the scientific process too. And then teaching them, as they go through each portion of the process, but also really to make it their own. Another trend in scientific inquiry, learning and teaching is to take it to the next step with claim, evidence, and reasoning in their scientific communication. And so it's teaching them, if they're going to write a report or have a conclusion, they're asked a scientific question, that they know how to make a claim, right? It's not an opinion. It's a claim that's then based on evidence, that they can point to something and say, the data from this date to this date show, and I know this is the case because of, right? And so they work through this CER process. So you're going to see that phrasing quite a bit come up more and more in the lessons as we have these essay questions. And sometimes we'll have, the rubric there that will show, here's how you score it for a correct answer. But with CER, sometimes there's not one exact, precise answer. It may vary based on the experiment or investigation that was performed and what evidence the students are pointing to. So they may get different results, right? And sometimes, as we see in real life with science experiments, everything doesn't always go as planned. And so one group may get a different result than another group. And, especially when we're dealing with these types of plants experiments, sometimes these plants, they have little minds of their own and do different things, and you'll get nodulation in one of them and not in another. And sometimes you just can't explain why, right? So we're still all learning through this process. On the platform, you will see this variety of lessons and content. And so we'll really be focusing on that scientific inquiry process as the primary sequence of lessons. But there's the content ones that will really go more in depth. And so you'll have the choice for teachers to be able to say, you know what, we're going to really go into, if you've got a more physics classroom, let's go more into the rocket science part of it, right? There will be a lesson on that, or more on the biology side with plant physiology, maybe even some genetics. Legumes in the nitrogen cycle, we just recommend those anyway, so the kids understand, well, what is a legume, right? Even the older ones can't always tell you what's the difference between a fruit or a vegetable or a legume, right? They haven't necessarily learned that. Tropisms is something that's new probably to most students. Middle school to upper school may be familiar with it, but unless they specifically studied it within plant science, they probably don't know what that is either. And then it's also just such a great opportunity to break out those microscopes and get them using scientific tools and learning how to do that. Even on the picture here with the seeds, I took that photo with just a little digital handheld microscope. So you can do quite a bit now with even minimal equipment. And the student kits come with a magnifying glass, which is really important too, because as you can see in the clover picture next to the ruler, those are nodules on the root, and they stretch from almost right at the base of the stem all the way down to the very bottom. You can get nodules in any place. It can be along that main taproot or any of the branching roots, and they're only about 1 millimeter in size. So especially when they're in the media, having that tool of the magnifying glass, even while it's in the ExoLab or in a bottle for a student kit, having that magnifying glass is a good thing to have on hand to look for nodulation. Because initially, when a branching root starts to form, it can kind of also start looking like a nodule. And so you want to pay attention to it and see, what kind of shape it takes. And then you'll be able to tell over a day or so, 2 days maybe, if it's going to turn into a branching root or have be one of those winning nodules. We talk with the students too about why we're studying food crop production in space. Why do we need to do this? And so we connect it to social studies and histories lessons back from those early explorers. Malnutrition was a leading cause of death, and the local indigenous peoples helped the new inhabitants survive by teaching them how to grow food. And so humans have to have food for energy, and the packaged products that NASA currently sends up to the International Space Station. It's great that you can send them up on a resupply mission, and they have those cargo shipments every few months. But eventually, the nutrients break down, and for growing plants, we emphasize that because it's a little hard to transport cows and chickens into space, and so it's more difficult. Though some researchers are exploring using insect proteins as a source beyond just plants. You can also go in depth, depending on your course, into the effects of spaceflight on human health, and then the role that plants play in counteracting that effect on the human body. And so this was a really helpful chart that kind of derived even from some of my graduate school work. And NASA is really looking towards the future, and so are we, about how can you use plants as that countermeasure. That nutrition plays such a crucial element in dealing with radiation even, right? So your different vitamin C, E, you have cardiovascular deconditioning, you have muscle loss, you have bone loss. There is no single countermeasure that's effective for these different conditions. And so they look at using a combination of measures to help humans survive, right? We are actually very fragile creatures. We have a very specific range of tolerance. And if we send up prepackaged food and the nutrients have broken down, we may get calories, but our bodies aren't actually getting what they need. And so plants are going to be a supplemental source for nutrition, but play definitely a crucial role in counteracting the negative effects of spaceflight on the human body. If we think out with a 3-year mission to Mars, given the way the planets have to align, the amount of food that is just required for those few members of a crew, 24,000 pounds, that's filling entirely 2 SpaceX Dragon cargo capsules. And so you can do some little math lessons with that. And the food would also need to be shipped ahead of time. They don't want to ship it at the same time. The thought process is they want it to get there and land on the surface, and they know it's there and it's made it there before they send people. And so now you're talking about even further out before the food's going to be eaten. And so that degradation, it's not only going to be the age, the nutritional decline, but the astronauts have to be willing to eat it. And if it does not taste good and the texture is not good, that's going to be pretty tough. Yes, they would probably go into Mark Watney kind of survival mode, but studies aboard the space shuttle missions as well as the ISS, they track their diets very meticulously. They use an app now, like on an iPad, to make sure that they are getting the nutrients they need, that they control their sodium levels, all of that. But there has been history of weight loss among astronauts because they were not eating enough. And that's another effect of space flight, of where it's like all your organs kind of shift up even. And so they don't always necessarily even feel hungry. And so if something doesn't taste good, on top of them, they're already feeling full, or they may have space motion sickness. They may, this could be a concern, because once they get to Mars, they'll have to be able to take care of themselves when they get out of that spacecraft. There will be no one there to help them out like there is today when they come back to Earth, and they will have to get out on their own. So during flight, as well as once they arrive, having that supplemental nutrition and food is going to be a necessity. And so we are teaching the Artemis and Mars generation. They will be the space farmers of the future for the moon, for Mars, and to deeper into the solar system. There's several great TV shows out there right now that even show greenhouses, right, on some of the moons deep into the solar system. And so we want these kids to really be inspired and realize that farming on Earth, has a certain persona or description, but a farmer of the future is going to need so many different skills, from robotics and computer science and chemistry and biology, and that it's going to be a highly skilled position. The hardware that's currently used on the ISS to grow plants includes the veggie system. I mentioned the advanced plant habitat. So each of those have a little bit different sensors with them. The APH is a bit more advanced, and they're able to monitor it a little bit differently. But the veggie unit has been awesome. They have 2 of those, and they have grown a number of microgreens and lettuce and radishes and some different crops. So here on the right, we have the ground control unit for ExoLab for the classrooms, and then the ExoLab Space Tango chamber that goes up to the ISS has to be a sealed box, because they don't want bacteria and such escaping on the ISS, right? And so we have to be completely sealed out, but they're still able to take images inside and use the sensors to measure luminosity, CO2, temperature, humidity, and then those images, when the box is, or the Cube Lab is plugged in, it's powered on, we get regular downlinks. We'll go just a little bit into the science, because some even teachers don't really know what they're looking for. This, it's a little bit easier to see the nodules. And on that left-hand side, this is a bean plant, a garden bean plant. And you can see the complexity of the nodules that have formed. And so this is a mutualistic and symbiotic relationship between these nitrogen-fixing rhizobia bacteria. Within the inoculants that you receive, it's often going to be a combination of 3 types of bacteria, but rhizobia is the primary one. And so in return, the plants are giving carbohydrates to the bacteria, while the bacteria is giving them nitrogen in a form that the plant can use as food. And so nitrogen is a limiting growth factor that can stunt plant growth. And so this is a biological fertilizer, and it's much more sustainable. So it's a win for bacteria, plants, humans, and the entire ecosystem. You can see on the right-hand side, this is a garden bean plant where there are no nodules. On this particular plant, when it had pretty much died by the end of the season and I pulled it up, I had a nodule or complexity of nodules that were several inches in size. So they can get really large on different legumes. And it's fun too to grow different legumes. That's another extension project. You could, as kids want to take it and explore on their own, take it to the next level. They may compare, what happens when you grow alfalfa versus clover, or garden beans or chickpeas. And that kind of is what we did during the Leguminaut Challenge. This shows some of a diagram of what's actually happening when the bacteria are introduced at the cellular level. The plant has to allow them in, and they come in through the root hairs. and you'll see on the clover plant very, very fine hairs on those roots. And so that's the access point. And if the plant is stressed, it won't let the bacteria in. It goes into self-preservation mode. It uses chemical signaling to say essentially yes or no. And so there's different, just like with all organisms, range of tolerance, right? If it gets too hot, if it gets too cold, access to water, all kinds of factors can affect nodulation. And so the question here that we're facing is this stress of spaceflight going to inhibit nodulation? And we don't have a conclusive answer to that question yet. That's why we're in part flying the experiment, right? Because what's the difference between what's happening on Earth versus what's on the ISS? Well, we're sending it up on a rocket, so it's facing some G forces, and then we're removing gravity. And so will the plant, will the seed and the seedling interpret that as too stressful of an environment and prevent nodulation? So that's kind of the focus of our study. The biogeochemical cycles are another great lesson extension throughout the entire project, particularly the nitrogen cycle. When I taught high school AP environmental science, this seemed to be the hardest cycle for students to learn for whatever reason. But I think it's one of the most fascinating cycles. They learn the water cycle typically in the younger grades, and there's some great songs about it. And even the carbon cycle, they can kind of get their heads around. But even though the atmosphere here on Earth is predominantly nitrogen, they just, I don't know what is in their brains, but they have a hard time. Part of it, I think, might be the vocabulary. When we talk about going from ammonification, nitrification, denitrification, they're like, what is all that? Those are some big words. So for the upper grades, you do want to emphasize that vocabulary. For the lower grades, we keep it simpler. But overall, what we want them to understand is the real emphasis on bacteria, right? We have these organisms within our soils that we have living dirt, right, here on Earth. And bacteria play this crucial role in decomposition and breaking down the waste and other plant material and removing the elements and minerals from that decomposed matter, and then turning it back into nutrients for plants to use. Then organisms like rabbits or humans, we consume them. But this special class of bacteria can kind of shortchange that whole process that would normally have to go through to have the bacteria and the legume form these nodules, and it's an immediate form, a direct source for the plant to grow. So, we're looking at project extensions where we really teach them about nitrogen, phosphorus, and potassium, those crucial elements within the soil, so that plants can really thrive and grow. But then what happens if we use chemical fertilizers instead? I did a project with my students where we did algal blooms in test tubes, right? And we got pond water from our campus source, and we fed it chemical fertilizer. And within a couple weeks, it was a complete algal bloom in the test tube. And so we have a huge problem here in the United States, particularly in Florida, in the Gulf Coast area, with a lot of runoff, right, from agriculture as well as septic systems, right, where you have these excess nutrients in the soil, and it causes these algal blooms that then leads to eutrophication of our waterways and polluted waterways. So we've had this cascade effect even here in Florida, where we've had a significant number of manatee deaths over this last year, and it's been traced back to excess nutrients in the water, which prevented the seagrass from growing, which meant that the manatees did not have a food source. So huge earth connection when we're able to show them that while we are going to be growing plants in space, the science that we're digging into is so important to then apply what we learn to solve problems on Earth. Plant physiology is really a fun lesson to do with them, as they can break out those crayons and markers, watercolors, and show their artistic skills, that A part of STEAM. So this is when we were doing the Leguminate Challenge, one of my students showed her knowledge of vocabulary of the plant physiology with this beautiful watercolor combination and drawing. So that's an important part as they learn from, we're going to be growing, from the seed stage, and it's not going to be on station very long, but they could keep their plants growing. And in this media, like, they can keep growing for months without having to add any water. And especially if they nodulate, they will stay living for quite some time. It takes a while for clover to get to the flowering stage, but it's not impossible. So they could even take them and replant them into soil and try to keep them alive, add it to their own gardens, or at home or at school. Tropism, so this is something that is really fascinating with these experiments, right? Because here on Earth, we take it for granted, really. But especially it's a very common one where, you put a plant in the window and it's going to bend towards the light, right? That's phototropism. And gravitropism tells us that the root's going to grow down and the stem's going to grow up. And we've done some things in experiments where, what happens if we put it in the dark, right? That's even a fascinating question that students can explore in the beginning. We normally put a dark period, a dark couple of days, because we want to simulate from the time that we've gotten on the rocket to before we get powered up on station. Sometimes that can be up to a week, right? And so if we have this period of dark and our plants start growing, what happens to them? Well, clover really likes to germinate fast. And so once it's in the media, it can germinate even within 12 hours. And you'll start seeing that taproot emerge, that radical emerge. And the root will be also sometimes confused and not always grow down. And the stem is looking for light. The leaves are looking for light, and it may become very elongated. And so if we have plants that start growing too fast in the dark, then they become very weak plants. They're very stressed, and it's harder to get them to nodulate in those conditions. And so we have to come up with some engineering techniques to, how do we prevent our red clover seeds from starting to germinate when it's going to be inside a box where you don't have astronauts who are going to start the experiment. So what techniques can we use to delay germination? And so that's been one of our challenges with ground trials over the last year, is to explore some of those ways. And so students can also come up with some good ideas. With gravitropism as well, you'll see that when we're on the ISS and we've removed gravity, it's in that microgravity environment, the root will really grow in any direction. But the leaves and the stem are very much attracted to the light, and they will bend pretty consistently and grow towards the light. When we get to our post-flight science with microscopy, you can do higher level gram staining techniques even with the upper level students. Students also have a false notion often about what they're going to see under a microscope if they've never used one or seen live cells. And obviously it depends on the level of magnification. But we have these diagrams, right, and models of cells that we often point them to in textbooks. But that's not what they're going to see, right? They're not always going to see all the organelles. And so we want them to be able to visualize and understand that sometimes you might see a cell wall and that might be it, right? That these are living organisms too. So the staining techniques can help them with some of that visualization. We start introducing to them this idea of why are we, really digging into this and exploring this, right? When we kind of ties back to Artemis, the moon, Mars, right? We have living soil here on Earth, so in many areas it's becoming depleted of nutrients, or it's becoming polluted with too many artificial fertilizers, right? And so when we get to the moon or Mars, it's dead dirt, and dead dirt doesn't grow anything anywhere, right? So on the moon and Mars, we know that the chemical composition will support growing some plants, but we've got some challenges, right? It doesn't have the microorganisms that the Earth soil does, and so we'll need to take bacteria with us. It has on Mars toxic perchlorates. And so what are techniques to remove that chemical signature from the soil so that it doesn't get into the plants and then humans would eat it? So it can be poisonous to the plant and poisonous to humans. That could simply be resolved by, water running through it, right? We've also got a lot of clay-sized particles in a lot of these, the regolith. No, you can do some things to help modify it and put vermiculite or perlite or other things into it to try and have some air circulation. Otherwise, these clay-sized particles really suffocate the roots. And a lot of the simulants that you get from different labs around the country, here I've gotten UCF as well as University of Central Florida. They have a great lab, a great simulant, but it is like a fine, floury type powder. And it's very hard to grow anything in that, right? And so we have to talk about with them particle size, the permeability, the porosity, this soil texture, pyramid. Great lessons for upper level students in particular, but younger ones can start learning some of these terms at least. So we have to deal with the nutrient deficiency, even thinking about, earthworms, right? What can they add to the soils? So we have a lesson on the platform that's an introductory lesson right now on growing in a simulated regolith. And we're looking at over the next year to kind of add that in as an additional experience of growing in your own soil, right? What will the red clover do? And let's do a global comparison with soils from Australia versus Japan versus Zimbabwe versus Florida. What are the quality of our soils when it comes to nutrient content? Good culmination opportunities for students to take everything they learned and do poster presentations. They can do science fair projects with it. You can either print them out or do it as digital. This was from one of my students, and she did it completely through a Google slide. A single slide, and incorporated everything that she had learned and worked on through the Leguminaut Challenge, and then presented it to her classmates. And here they had to advocate of why they thought a particular legume should be our candidate. So she had chosen lentils. So on the platform, I know you've seen the experiment side a bit. So this is what it looks like when on the left you have your ExoLab, that's your ground, and then on the right you would have the one that's on the ISS. I just popped this in there today because we're doing new ground trials, and so my colleague Michael is in New York, and he has a ground station that we're all pairing to at the moment. So these are both on Earth right now. But you can see the kind of comparison images and data that you would get. Now, I have a little fun thing going on with my lab right now too. It's turned on its side, and I did that specifically because we had a challenge in the last mission with the media rotating within the tube. And so we're trying to find mitigation measures to prevent the media rotation. So you can see on this left-hand image, that right-hand tube, the media is still rotated. And I was trying to use a combination of BioStraight felt and even just Elmer's glue to try to hold the media in place. Yet gravity here on Earth is still affecting it. And so we get that media rotation. So that's still a work in progress, you could say. And while we're going to be in the microgravity environment from the time that we are assembled, right, by Space Tango into the Cube Lab, as I said, it could be a week time frame going from packaging, preparation of the experiment, getting it transported to the rocket, loaded up. It is in gravity all of that time. It is going to be shaken, it is going to be jostled, it's going to be turned up and down, sideways, who knows? And then it's going to be subjected to G-forces and as it launches, and then eventually reach the station within about 7 days is what it typically is. So we have to really make these strong enough that it can take all that stress in preparing the science protocols. So we're still working on refining some of those. We are going to send up 3 test tubes in the flight experiment with 5 seeds each. Given the size seeds we've got and only having about 30 days on station, the plants will stay very small and they can achieve nodulation. We've had on Earth nodulation happen in as few as 8 days. Typically the average is more like 17 to 21. And so we're definitely within our time frame to be able to achieve nodulation under what we would call normal conditions. And so then you can also see the data comparisons for CO2, humidity, temperature, and light. And you'll also be able to compare to other labs around the world and look at what their plants are doing and how they've set theirs up. And this is our Bitmoji magnitude team of investigators and everyone that's been working on our plane experiment and getting us to space.
So I really hope you can get involved in the ExoLab project. It is such an awesome opportunity to really engage kids in true space science research, citizen science, and really spark their imagination for potential space science careers. There are so many opportunities, especially as we look towards 2030 and the space economy that's happening in Australia right now. So if you want to know about the ExoLab, please reach out to me at Physics Education, so Ben Newsom at physicseducation.com.au, or reach out to any of the team through the office. We will be able to let you know all about how the ExoLab works and more importantly, how you can get engaged and get one for yourself and to get the students involved in their own growth experiments. It is so very, very powerful. And I've heard firsthand in many live webinars with kids about what they've been doing and how much it's changed their perspective on real space science. So hope to get in touch with you.